You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is November 11th, 2024. Our topic today is Abbas Chapter 5, Antibodies and Antigens. This is presented by Dr. Nikita Raje. She's an Associate Professor of Internal Medicine Pediatrics and is the Chief in the section of Allergy Immunology.
Hope you guys had a good weekend and had some time to go over chapter 5. We're going to be talking about antibodies and antigens from Abbas today. And I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City. Today is November 11, 2024, and my email is listed in case you have comments or questions. So let's get started here. Let's see if I can move this. All right, few of my disclosures, nothing relevant to the talk today. Our objectives for today's lecture are to expand the understanding of antibody structure, synthesis, assembly, and expression of immunoglobulins, antibody binding of antigens, and then we'll go over structure and functional relationships in antibody molecules. Couple of pretest questions here. Which of the following is a correct description of the basic symmetric core structure of an antibody, immunoglobulin G antibody? Let you go over those options. And then next question is, an antigen binding site of an IgG antibody molecule is composed of which of the following? All right, we'll come back to those. Let's talk about antigen binding molecules. So antibody was named because it can bind to antigen, but apart from that, there are other molecules that can bind to antigens as well. We know and we have discussed T cell receptors, and those can also bind to antigens. So keeping in mind that there are several similarities in the structure of these molecules, such as the antigen binding site in the immunoglobulin is made up of 3 CDRs, the hypervariable region. They are present in the heavy variable chain and the light variable chain. Whereas for TCR, they are in V alpha and V beta domains of the TCR. So that's the part of the immunoglobulin or TCR that binds to the antigen. But there are also some differences, such as immunoglobulins can bind to various different types of molecules, such as proteins, lipids, polysaccharides. They can also bind some chemicals, whereas TCR is very specific in what it binds to. One, it needs the antigen-presenting cells that can express a peptide bound to its MHC chain so that it can present it on the surface so that it can bind to the TCR. The immunoglobulins are known that they can actually bind to linear or conformational parts of the antigen. These parts are the parts that come in contact with the antigen binding site are called determinants or epitopes, and those determinants can be linear or they can be folded or what's known as conformational, and doesn't matter which one. However, TCR can only bind to linear determinants, which means it's a chain of peptides and that's about it. It's only a few molecule or a few amino acids long based on what can fit into the MHC molecule, which we have talked about in the past. So let me know if you need a refresher on that. And then the affinity of antigen binding is different, so that for immunoglobulins, the antigens can bind rapidly, but they can also come off at a variable rate. However, the TCR has a slow on rate and off rate, which means it binds slowly, but it can also take time before it can get detached from there. So let's talk about the antibodies. So these are either membrane bound, so they're bound to, say, B cells, or they are secreted. And the classes of antibodies are based on the heavy chain of the immunoglobulins. Based on those, the classes can be IgG, A, M, E, D. And the amount of antibodies that is released every day is about 2 to 3 grams. Interestingly, two-thirds of that is immunoglobulin A. So the most abundant immunoglobulin in our body is IgA. But not all of that is in the circulation. So the most abundant immunoglobulin in the circulation might be IgG. The effector functions of these antibodies can include several different ways where they work with to restrain the infectious agent or the antigen it comes in contact with. It can neutralize the microbes or the toxins, which means it will not let it enter any other cell. There is activation of complement system. That's the classical pathway of complement system. And then these can tag the pathogens for phagocytosis. So they opsonize pathogens for phagocytosis. They also take part in antibody-dependent cell-mediated cytotoxicity, along with NK cells and antibody-mediated mast cell activation that can lead to allergic processes. The structure of antibodies is very specific. It has 2 light chains and 2 heavy chains. And these 4 chains come together to form the variable region and the constant region. So antigen binding region is the variable region, and it's important for the specificity and the diversity of antigens that can be recognized by that antibody, versus the non-antigen binding or the constant region is, as the name suggests, it's constant, it's not variable, but it's important for the effector function. That's what determines the classes of the antibodies, and also is responsible for all the physiochemical properties which differ among these classes of antibodies. So more about the structure here. Let's go into the detail of what these chains look like. So each chain of immunoglobulin has repeating units called immunoglobulin domain. So immunoglobulin domain is a structure with 110 amino acids that's laid out in 2 layers of what are known as beta pleated sheets. So they are sheets that lie on top of one another, and they go in each of the layer has these strands that go in anti-parallel direction. And these layers are connected to each other by disulfide bridges. The adjacent strands are connected by short loops that are called CDRs. And the amino acids that are present in these regions of CDRs is the most variable. That provides that diversity of antigens that antibodies can bind to, because each antibody is specific to a particular antigen, right? So the diversity in the amino acids present in these chains tells us about the immense diversity of antigens that these antibodies can bind to. So again, beta pleated sheets, and each of them have strands of anti-parallel polypeptide chains. The chains are bound to each other by the short loops called CDRs, and both the layers are bound to each other by the disulfide bridges. So immunoglobulin domain is this structure. It doesn't matter where it is or what molecule it is on. So the immunoglobulin domain can be present not only in the immunoglobulins but also in other molecules. So all the molecules that have immunoglobulin domain structure in them belong to what is known as immunoglobulin superfamily. So, of course, the immunoglobulins themselves belong to the immunoglobulin superfamily, but there are other molecules such as TCR, class 1 MHC, CD4 molecule, CD4 surface marker on T cells, CD28, ICAM. All of these have this structure that you see that's common to all of them. That's called immunoglobulin domain, and they all belong to immunoglobulin superfamily. So when we come back to our light chains and heavy chains, we know that the light chain, there are 2 light chains and 2 heavy chains. The light chain has two regions in it, the variable region and the heavy region, the constant region. And then the heavy chain has one variable region, and then it has either 3 or 4 constant regions in it. So the antigen binding site is just formed by both the heavy and light chain regions that are the variable regions. Each antibody has 2 antigen binding sites. So there are 2 variable light regions and 2 variable heavy regions that interact with each other to form these 2 antigen binding sites. The effector function is dependent on that constant region, the CH chains, right? And the classes are determined by these chains as well. In between the constant regions of the heavy chain, there is a hinge that helps in the flexibility of the antibody. And we'll look at that in a minute here. And of course, these chains are interacting with each other by covalent and non-covalent interactions to be able to have a stable structure. As you see, there is a difference between the secreted immunoglobulin and the membrane immunoglobulin. The membrane immunoglobulin will have a tailpiece that's different, long, and it can insert into the plasma membrane of the B cells where they are attached to. Whereas the secreted form does not need that long tailpiece, and it has that small piece that helps it to stay stable, but then it's secreted and does not need binding with the plasma membrane of B cells. All right, so to summarize, 2 light chains, 2 heavy chains, each of them having variable and constant regions. The constant regions in the heavy chain can be either 3 or 4, and that determines whether it's membrane bound or secreted, and then the effector function is all dependent on the constant regions, whereas the antigen binding site is provided by those variable regions. So the structure of the immunoglobulin was understood because of the experiments that were carried out on rabbit immunoglobulin. And the 2 ways or 2 enzymes that were used to study these rabbit immunoglobulins were papain and pepsin. And so when papain is used, the break in the structure is before the hinge. And so there are 2 pieces that have the FAB region. The antigen binding fragment is what it's called, FAB, versus the hinge along with the other constant region forms the FC region. If instead of papain, pepsin is used to proteolyze that immunoglobulin, it acts below the hinge so that it produces this one piece that has 2 FAB regions, and then this rest of the constant regions just disintegrate into peptide fragments. So again, these are board questions. They are in red, so memorize that. Easy way to remember is that papain has 3 vowels in it, so there are 3 pieces of it, versus pepsin has 2 vowels, so it makes like 2 parts of it. One has both the FAB regions, and then rest are peptide fragments, just for memorization. But pretty much kind of you have to realize why that happens. It's either the break is above the hinge or below the hinge.
Dr. Raje, quick question. Like, I know that this is it just comes up on boards, but like, is there a reason why it's important to know this? Just out of
No, it's so earlier when you have to realize that this was all experimental and someone studied these molecules, the structure, because the structure of immunoglobulin, if it was not known, I don't, science develops that way and progresses that way, that it depends on all these discoveries that were made, right? So somewhere along this was done, but no, not in my mind. I cannot think of a clinical reason for us to memorize that.
And then just another thing for when it says FAB and then like is it just called FAB? Is that just
It's an antigen binding fragment.
Oh, okay, I see. Okay, got it.
FAB, and then the constant fragment, which is the C frag, C region.
Okay, thank you.
All right. We are diving even deeper into that structure for the variable regions, which are the important part that determines the specificity and the diversity, right? So thinking about those CDRs or those short loops that connect those strands of polypeptide chains. And among them, there are these three CDRs, CDR1, CDR2, and CDR3. And they stand for complementary determining regions. And these CDRs are responsible for the most variable parts of the antigen binding sites. They may include some conserved portions, but CDRs are responsible for that antigen binding. So the most variable, these are also known as hypervariable regions, right? These are confined to three regions of both the variable light and heavy chains CDR1, 2, and 3. And these regions have 10 amino acids in them. And they're the most, so what's the most variable CDR? Second years.
Is it IgM?
No, I'm talking about the CDR1, CDR2, or CDR3. Which of these is the most variable region that determines? So all three of them are called hypervariable regions, but one of them is responsible for the most variability.
Yeah, just from last year, I think it was CDR3.
Yep. So CDR3, again, really important to memorize. All these three regions are, remember, all three are CDRs, which means they are hypervariable, but the CDR3 is the most hypervariable. Constant regions. So we talked about the variable region that gives the specificity and the diversity. And now we talk about the constant region, or the FC region, that determines the classes, or the isotypes is what they are known as. GAMED, G and A, we know have subclasses, so G1 to G4, A1 and A2. The constant chains, depending on the class, depending on the chain that's present, determines the class, which means if it's a gamma chain, it stands for IgG, alpha chain stands for IgA, mu for M, and epsilon for E. In IgG and IgA, there are three gamma or alpha chains, versus for M and E, there are four chains. As we talked about, that determines the class, also determines the function, and the way it determines that is depending on the class, the cells and the proteins that can interact with that region is specific. So for example, if it's IgE, it may be interacting, it's able to bind to, say, mast cell or eosinophil or basophil, whereas if it's IgG or A or M that helps in phagocytosis, it can bind to macrophages or things like that. So basically, it determines the cells that these immunoglobulins can interact with. And because they can determine the cell or the protein that they can interact with, they also determine the type of effector function, what it does, right? Whether it's a mast cell and what that function is versus a macrophage and what that function would be. Looking at these isotypes, IgA has 1 and 2 subtypes, and the half-life is, again, for all the immunoglobulins except for G, is pretty short, few days. For IgA, it's about 6 days. This is present in secreted form, mainly as a dimer. What does that mean? There are 2 molecules of IgA, which means there are 4 antigen binding sites in that whole structure, and those 2 molecules of IgA are bound to each other by something called J chain or joining chain. It's important for mucosal immunity, as we know. IgD is has a half-life that's very short, about 3 days, present as a monomer, and it's present as an B cell antigen receptor, right? So the B cell antigen receptor, if you can recall, we talked about how there is IgM on the surface and then there is IgD on the surface as the maturity, with the maturity of the B cells. IgE, again, really short half-life, about 2 days. It's a monomer, and we know that it helps in defense against helminths, parasites, and also is responsible for allergic issues. IgG, G1 to G4 are the subtypes. It's mostly a monomer and has multiple functions. Helps in opsonization, complement activation, ADCC, neonatal immunity, and it also provides feedback inhibition of B cells, so it helps in regulation. IgM, it's a pentamer. So pentamer, which means 5 molecules, which means 10 antigen binding sites. And all of those are bound to each other using that J chain. What's the role of IgM? It's the receptor, right? It's the naive B cell antigen receptor. Presence of that is required on a B cell, or really, is the B cell antigen receptor itself. And then also it fixes complement. So that's an important function as well. We talked about the variable region, we talked about the constant region, and in between those two is the hinge region. It's between CH1 and CH2. It's about 10 to maybe more than 60 amino acids in length, and it's responsible for the flexibility of the antibodies. So say it's an antigen that has repeating epitopes, and 2 antigen binding sites of the antibody can bind to those 2 epitopes on the surface of that microbe. Well, sometimes that can be widely spaced or they can be closely spaced, and the hinge helps to either widen or narrow the regions between the 2 antigen binding sites so that it can bind to the determinants or epitopes on that antigen. All right, so light chains are 2 different classes, kappa and lambda. In humans, 60% of the antibodies will have kappa light chains versus 40% have lambda light chains. That ratio can change with B cell lymphoma, and that's the reason it's one of the tests that you can use to look for lymphomas. All right, we talked about monomers or dimers or pentamers. We know that IgE and G are monomeric versus IgAs can be dimeric, or IgM can be either pentamer, which is the most common form, but IgM can also be a hexamer. With increased number of antigen binding sites, the avidity, even if the affinity is the same, the affinity depends on the interaction of one antigen binding site to its antigen. Compared to that, the avidity is a overall strength of that interaction, including all the molecules that are present. So in case of IgA and IgM, the avidity can increase. We talked about the secreted and membrane forms, how the tailpiece is longer in case of membrane immunoglobulin versus the secreted immunoglobulin. Talking about differences in antibodies, we know that if serum antibodies are introduced from one species to another, then they will be seen as foreign, and the immune system will mount an immune response. The antibodies are typically formed against the C region of those antibodies that are introduced, right? And that causes serum sickness. There are different types of antibodies, such as anti-allotypic antibodies, which means the differences present in antibodies from different individuals of the same species. Those differences are pretty small, and these are called allotypic typic antibodies. Anti-idiotypic antibodies, which means there is even smaller differences between the variable regions concentrated just in the CDR part that constitutes the idiotypes of antibodies. All right, so two different species, same species, different individuals, and then same individual, but there is a difference between the variable region. So, and either those are anti-allotypic for individual differences versus CDR differences idiotypic. All right, so what are monoclonal antibodies? So we know that we use a lot of monoclonal antibodies in clinical medicine. What are these and how are they produced? So the antibodies of any desired specificity produced by immortalizing the individual antibody secreting cell from an animal immunized with a known antigen. So let's look at this figure. This was introduced by Georges Kohler and Cesar Milstein. These are called hybridomas. So if we look at this here, say a mouse is injected with antigen X, and then the antibodies are collected from the mouse spleen that gives antibodies against that antigen X. So that we'll call them anti-X antibodies. Those cells or cells that produce antibodies can be fused with myeloma cells. So myeloma cells are cells that can produce lots of antibodies myeloma cells are the ones that are specific in terms of they can produce they can multiply a lot but not produce antibodies, but they are long living, versus the ones that are coming from the mouse are specific to that antigen. And so when you fuse them, you get fused cells. So the unfused cells will be taken off. And so the medium that is added to this experiment, hat medium, takes away all the unfused cells, and the ones that are fused cells will have the properties of myeloma cells, so that they are long-lived, and then they can continue to produce the antigen that the specific cells were to get taken from the mouse. These are called hybridomas, and they can produce monoclonal antibodies. So the antibodies coming off a hybridoma are specific to that particular antigen. This whole process is used and has several different types of applications, as we know. For a start, we use these to help identify different types of cell populations, right? So we use clusters of differentiation to define if a cell can bind to a particular monoclonal antibody. So for example, an anti-CD3 can be used to identify cells that can bind to the CD3 cells that possess CD3 on their surface, and that's how we define our T cells, for example. So each of these individual cell populations can be identified by specific monoclonal antibodies that can bind to it. They are used in immunodiagnosis, detection of particular antigen or antibody in body fluids. We use serology all the time for various reasons. We can also use identification for tumors using some monoclonal antibodies against certain proteins. We use monoclonal antibodies against cells involved in pathogenesis so that we can regulate those cells or target those cells in treatment. And then we can also do functional analysis of cell surface and secreted molecules using these monoclonal antibodies. So when these monoclonal antibodies are discovered or kind of invented, we have a particular type of nomenclature that is followed. And so we'll go into the details of how these are named. One of the limitation of monoclonal antibodies is that you can have human anti-mouse antibodies. So if they are coming from mouse, then the immune system can give rise to antibodies against those monoclonal antibodies and cause problems. So because of that, genetic engineering can be used to obtain cDNA from the hybridomas and cDNA from the myeloma protein to form hybrid genes. And that can give you genetically engineered antibodies as well. So depending on how much of the antibody or monoclonal antibody comes from the mouse, the suffix of the that is given to the monoclonal antibody types is different so that you know what type of antibody you are dealing with. Such as if it's all coming from the mouse, which means it's a murine antibody, then it has a suffix OMAB. If only the variable regions are coming from the mouse and the rest of the antibody is genetically engineered, it's called chimeric. Anyone know what the suffix is for that? It's called ZMAB. And then humanized, which means not even the whole variable region, it's just the CDR that's coming from the mouse. Rest of the monoclonal antibody is genetically engineered, and that's called ZUMAB. And then human, so which means the whole antibody is from human, and that's UMAB, right? So some of the examples, I'm gonna say the name, you tell me which category these belong to. Miromonab.
Murine?
Blinatumomab.
Murine as well.
Rituximab.
Chimeric.
Chimeric.
Infliximab.
Chimeric.
Chimeric.
Omalizumab.
Humanized.
Human.
Humanized. So Zumab. And then Adalimumab.
Human.
Human. Benralizumab.
Humanized.
Alivizumab.
The
So you
same
get the idea.
humanized.
So you get the idea. So thinking of the name, you would know how much of the antibody is coming from the mouse. Here is an example of different kinds of monoclonal antibodies. These monoclonal antibodies, a lot of these are being used for different reasons, such as monoclonal antibodies against alpha-4 integrins, used in multiple sclerosis and Crohn's disease, CD20 monoclonal antibody for depletion of B cells. We know the rituximab is one of the examples, but there are several others that now we have that are used for treatment of autoimmune diseases or malignancies. IgE antibodies used in allergic disorders, TNF antibodies used for various inflammatory processes and rheumatological conditions, Crohn's disease, rheumatoid arthritis, psoriasis. IL-17 is used in psoriasis. Similarly, that's just a list of things that's mentioned there. I'll let you guys read. All right. Thinking about these immunoglobulins, where they are synthesized, we looked at the structure, we looked at the function. and we look at where it's synthesized. So as we know, it's all proteins are synthesized on ribosomes. Same thing with the immunoglobulin molecules. They have light chain and heavy chains. Both of those chains are synthesized on the ribosomes, and then they are in the endoplasmic reticulum. They are folded. These proteins have to undergo folding with the help of chaperones, and then once that's completed, they are released onto the Golgi apparatus, and here they are modified further with some carbohydrates. The carbohydrates are modified there, and then these proteins or chains are packed in vesicles so that they can go to move towards, those vesicles are moved towards the plasma membrane. If these are membrane bound immunoglobulins, they will be anchored to the surface of the cell, or if they are secretory, they will be transported out of the cell. All right, so we talked about antibodies. Now we want to think about the maturation of B cells in terms of how the immunoglobulin plays a role, especially IgM. We also talked about IgD. So based on the stage of maturation, and I think this is a really, really important figure to memorize, the information to memorize, because the stage of the maturation also decides what's on the surface. If there is a break in that maturation in some disorders, then you know, depending on what you are looking at and what markers are present, you would know where the maturation stopped, was arrested, right? So in stem cell, there's no immunoglobulin produced. In pre-B cell, the mu heavy chain is starting to be produced, but it's in the initially in the cytoplasm, and then later it is present on the surface, mu heavy chain, in conjunction with a surrogate light chain. So it's a pre-B cell receptor. There is no IgM on the surface. This is a mu heavy chain along with a surrogate light chain. And that has to be tested again so that the B cell survives. If it undergoes that testing and is able to survive, then it moves forward to becoming a immature B cell when IgM is present on the surface. When the B cell is released into the circulation from the bone marrow and in the periphery, the B cell undergoes further maturation, and when it's mature, it has IgM and IgD on its surface. So that's a fully mature B cell. It has to have an IgM on its surface and IgD on its surface. And then when the B cell gets activated, it can actually transform, so differentiate into a plasma cell, right? A plasma cell is the one that's gonna be the one that's gonna release antibodies. The activated B cell will have low rate of immunoglobulin secretion, but it undergoes isotype switching, heavy chain isotype switching, and affinity maturation in the FAB region. And then the plasma cell has high rate of immunoglobulin secretion and little or no membrane immunoglobulin on its surface. So the surface markers or the markers in that cell will determine or can help recognize what stage of maturation that cell is at. So really important information there. All right. We looked at the half-life, few more details about the half-life. This is a very short half-life for most immunoglobulins, like IgE for 2 days, A for 3 to 6 days, IgM for 4 to 5 days. Compared to those, IgG has a long half-life, 21 to 28 days. IgG3 among these has a shorter half-life. And so how does IgG end up with such a long half-life if the structure is, most of it is pretty similar, right? And the reason for that is it's binding to a receptor called FcRn that stands for neonatal Fc receptor. So Fc is the constant region. Remember, that's the one that interacts with proteins and different cells. So the neonatal FC receptor is found on various cells like endothelial cells, macrophages, and it binds to the immunoglobulins. So see, if you look at the figure here, the pH in the blood is 7.4, but when the proteins, including immunoglobulins, sorry, I can't get here. So immunoglobulins are taken into the cell, so phagocytosed into an endocytic vesicle. Inside the endocyte, the endosome, the pH is acidic, and because of that, the antibody immunoglobulin G can bind to the receptor FcRn. When it's bound, it escapes it escapes the degradation in the lysosome. So the proteins that undergo degradation in lysosomes are the ones that are not bound to FcRn. So immunoglobulin, because it's bound to the FcRn, it escapes that degradation, and then it can undergo recycling, which means the endosome is going to take it to the surface of the cell, and where the FcRn, because the pH is again neutral, it's going to be released into the circulation. Because of this, the immunoglobulin stays in the circulation because it's initially taken in and then recycled. It stays in the circulation for a longer time. This property is really helpful because you want some of the drugs that we give to have a longer half-life. So some of the drugs can use this property if they are bound to the Fc region of immunoglobulin. So that's used in certain medications that are called fusion proteins, and they have the specificity of the drug, like TNF receptor, and they are bound to the Fc region of the immunoglobulin G that can bind to the FcRn and can give a longer half-life. And these drugs end in the name sept. And so it's like TNF receptor immunoglobulin, etanercept is one of the examples. CTLA4 immunoglobulin is another one that's called abatacept. And so these fusion proteins are used in clinical, has clinical applications. All right, we talked about antibodies. Now we talk about antigens. So we know that antigen is a substance that can specifically bind to either an antibody or the T cell receptor, which means antigen-binding molecules, right? Among these antigens, some of them are capable of stimulating immune response, which are called immunogens. Some antigens are smaller, so they do not stimulate the immune response, but they can bind to the antigen-binding sites of antibody or TCR. So these are called haptens. They can bind to the antigen binding sites but cannot produce an immune response. Those are called haptens. Carriers are large molecules that can be bound to hapten so that once conjugated, they can together give rise to immune response. And then the portion of the antigen that binds to the antibody is a very small portion of that overall antigen, and that's called determinant or epitope. So those terms are interchangeable. So let's talk about these determinants. Give me 1 second here. All right, so determinants are, depending on where they are located or what part of the protein is the part that exactly binds to the antigen, these can be either conformational epitopes or linear epitopes, which means say a protein is folded onto itself to give a three-dimensional structure to it or conformational shape to it. Some of the determinants are present when there is that conformational change. When the protein is folded, that structure gives rise to the epitope that can bind to the antigen binding site. But if that protein is denatured and it becomes a linear chain, then that epitope is lost because it loses that structure that can bind to the antigen binding site. Similarly, some are linear to begin with, where if there is denaturation of that protein, it doesn't matter because that linear portion is still present in that structure no matter what. So those are the two different types of epitopes that you need to know, but then you have to realize that sometimes the determinants can be overlapping or non-overlapping, which means that and then there is something called allosteric effect. So the binding of an antibody to an antigen causes a conformational change in the structure of the antigen, influencing binding of a second antibody to another site, which means there is no steric interference. Steric interference means that there is such an overlap that if it binds to an antibody, another one cannot bind to that place. But allosteric is just that the conformational change that is produced by binding of an antibody to antigen that ends up in such an structural change that the second antibody cannot bind to that site. All right, and then the last one is neoantigenic determinants. These are epitopes that are produced because of alteration in the structure, which means that initially, whatever there is, whatever way the structure is for that protein, the epitope is not present. But because it underwent some change, there is an epitope that's produced that can bind to that antigen binding site. That's neoantigenic epitope. And so there are times when, this is one of the theories, for example, in allergen immunotherapy that we use, that when these extracts are used and some epitopes can change depending on what extracts you mix, there can be new antigenic epitopes that can be released that can also help with immunotherapy. All right. We did touch upon this a little bit earlier. So the affinity is the strength of the binding between one single combining site of an antibody and an epitope of an antigen. In case of monovalent antibody, the antibody can only have two binding sites that it's going to bind to. The affinity is just that one antigen binding site with its antigen. The dissociation constant is a measure of how easy it is to separate an antigen-antibody complex into its constituents. So smaller the dissociation constant, which means that it's stronger or has higher affinity, which means there is lower concentration needed for complex formation. So higher affinity means smaller KD, right? And then avidity is the strength of attachment of all the available antigen epitopes to antibody binding sites. And then you have learned this a lot in medical school. When there's a lot of antigen excess, there are smaller complexes formed. And there is a lot of antibody excess, same thing, small complexes are formed between antigens and antibodies. But there is a zone of equivalence where most of the antigens can bind to the antigen binding sites to form a big complex. When you think of these antibodies, they have certain properties that are important for the function of the antibodies. So the first one being specific, which means the antibodies are specific for a particular antigen. There's a lot of different, the next property is diversity, where there's a lot of different binding sites and different types of antibodies that can bind to multiple different antigens, and that provides the antigen repertoire that our immune system can have, which means there is a lot of diversity because of the different specific antibodies. But to make it a little complex, sometimes the antibodies can bind to an antigen that has similar structure even though not identical, and that's called cross reaction. So remember, there are FAB regions and FC region. If there is mutation, somatic mutation in the FAB region, then the affinity can change because the way the antigen can bind to the epitope, antigen epitope can bind to the antibody will change, and that's called affinity maturation. The goal is to have antibodies with higher affinity. That's called affinity maturation. Compared to FAB region, if when it comes to the FC region, that determines the effector function, and that determines what cell it can bind to. So if an IgE can bind to the FC region of IgE can bind to the FC receptor on mast cell, for example, when it's bound, it can cause degranulation. IgG can bind to complement, and then IgA can bind to mucosal antigens, and so on and so forth. We did touch upon class switching. Well, not today. So class switching is where the antibody can switch from one class to another, which means typically the initial antibody is IgM subtype and then or isotype, and then once that IgM can change the FC region or the constant region, it can switch from IgM to GA or E, depending on the one that's needed for that particular infection. That happens because of the gene segment for the constant region that can switch. Right. And then we know that the tissue distribution of each of these antibodies is different comparing IgA in mucosa versus IgG, which is more in circulation, IgM, which is bound to B cells, also in the circulation. IgE, again, can be in various different regions. All right, so that's the end of all the slides. Questions at this point before I go on to discussing the questions that we had at the start of the talk.
Yeah, Dr. Raje, for affinity and avidity, it's always been a little bit confusing to me. Could you give us is there any practical scenario where we would want to order avidity testing?
So clinically, we don't have affinity avidity testing yet. I know there are a couple of labs that provided it, but I don't know how valid those tests are. So we are not using those kinds of testing because that would be ideal, right? So when you think of B cell function, B cells, and you have to do testing for B cells, then either you can get quantitative testing looking for B cells, right? Whether they are present and enough in number or not. When it comes to qualitative testing, you think whether B cells can produce antibodies, and that's a good functional test, which means you can just check antibody levels. When it comes to the antibodies, that's your quantitative test, whether they're present or not. But the functional test is whether they can make functional or specific antibodies or not. For that, we use vaccine titers. So vaccine titers are a functional test for the function of immunoglobulins, right? Similarly, immunoglobulins are functional tests for B cells, but it's still a quantity. And when you think of antibodies, immunoglobulins are a quantitative test in terms of whether they're present or not. Same way, when you think of functional or specific antibodies, you can look at the number of antibodies, and that can be a functional test for your immunoglobulins. But when you think of it, it's a quantity of immunoglobulins that's present. Is the tetanus titer present or not? Diphtheria titer present or not? If it's pneumococcal titers, you are looking at various different serotypes and whether they are present or not. But it doesn't tell you about anything about whether they are able to whether they have the good affinity or not, whether they have the avidity or not. And that test is clinically not available at this point, or if it's available, it's not a validated test yet. So I don't know if I can tell you where we use it, but it's a concept because I definitely have had patients where you have the B cells, you have the immunoglobulins, you have the specific antibodies, and you know that there is still an issue that something that you are not testing, and you can see some of these patients having frequent infections to the same kind of uh, microbes, and you wonder if those antibodies are functional or not. Not that I have tested them for avidity or affinity, but I have definitely commented on it saying, at the end, we have quantitative information and we do not have that qualitative information. And that's likely a patient that may benefit from immunoglobulin infusions because those infusions would have functional antibodies.
So you would just have to suspect that even though their titers, even though the amount of their titers, even though the level of the titers meets the threshold, that they're not functioning the way that they should be.
Correct. You have to have, that suspicion. You have to be able to justify, when it comes to treatment, you have to be able to justify the use of some of these products.
Okay, got it.
All right. First question here, which one of the following is correct description of basic symmetric core structure of an immunoglobulin G antibody?
So should be too heavy.
D. Yeah, D.
All right, 2 heavy chains, 2 light chains is correct. An antigen binding site of an IgG antibody molecule is composed of which of the following?
This one should be D as well. The 6 hypervariable, 3 heavy and 3 light.
That is correct. 3 heavy and 3 light, 6 hypervariable loops is the correct answer.